No, I'm gonna leave. I can't be showing this on my game.
Good day, everyone. I hope you're all good.
Hey, Joe. How are you?
Hello, Professor Prahlad.
Hi, Joe. How are you?
I'm good. Thank you. Professor K is also here with us. Hello, Professor K.
Hi.
Morning.
Hi.
Hi, Professor Prahlad.
Hi, how are
Seeing you.
you?
It's been a while. This is our
Oh,
second time.
yeah. Yeah, it's
Oh,
good to wrap up the session now, so. Mhm.
yeah. Oh, by the way, there's a typhoon in my area. My connection is not very reliable
Oh,
today.
Yeah,
I hope you're okay. So, yeah.
yeah, same here. We have a super typhoon here in the Philippines, and this is actually the strongest typhoon recorded
Yeah.
this year. So, yeah, I hope everyone is safe and sound. All right. I guess some students are already here.
Hmm.
Okay. Professor Prahlad, would you like to start in 1 minute? We'll wait for
Uh,
1 more minute.
yeah, we can wait for a minute or so. Usually Joe used to start, so okay. So she's not going to be
All right.
here, it seems. Okay. Yeah,
Okay.
well, yeah, we can start actually. So
Okay. All right.
I think the
So,
students are here. So yeah, welcome to the thesis defense, the 2025 summer session. It's yeah, we've had a great session, and a lot of students are actually on the verge of submitting their review articles. So that's great. So today they are going to tell us exactly what they worked on during the summer. So we can start with maybe, I don't know, alphabetically going. Akhil is going to be the first one,
yes,
right? So,
Professor. I actually have here
uh,
a list. I can call their
oh, you
name.
do? Okay, yeah. So you can go ahead and call the names.
All right,
Yeah.
Professor. Thank you. Once again, good day, everyone. On behalf of the Gifted Gabber Summer Research Program, I would like to extend a warm welcome to our research students, parents, and of course, our professors. A special acknowledgement goes to our esteemed research professor, Professor Prahlad and Professor Kay, for their invaluable guidance and expertise in shaping the research projects that we are about to witness today. So today marks an important milestone in our students' academic journey, and the thesis defense not only showcases their hard work and dedication, but also their ability to think critically, present clearly, and engage meaningfully with scholarly feedback from our professors. So we will now proceed with the research presentation. So each student will present their city, after which we'll open the floor for comments and, of course, questions. I'd like to remind our students that after the presentation, you must post one question in the chat box, and our presenter will be answering those questions later. So you are all expected to post questions, all right? So this one ensures that there's an engagement and of course there's collaborative learning during the thesis defense. So let us now invite our first presenter to share um, his work, her work. Okay. So of course, let's give the floor to Ananya. Ananya?
Hello.
Hello.
Should I just share my screen and then I can present?
Yeah, sure, go ahead.
Okay.
And I'd like to
Mhm.
request our presenters to kindly open your
Oh,
camera
yeah.
during your presentation.
My presentation did end up kind of long, so I might go a little bit like quickly through it, but my topic that I was working on is bacteriophage therapy and how it's used to solve antibiotic resistance and how it's sort of turning out to be a double-edged sword currently in medicine. So currently antibiotic resistance, which is the ability of bacteria to evade the effect of antibiotics, it's becoming like a growing problem across the world. As you can see, this graphic kind of shows like the deaths that are happening across different continents, and it's responsible for a large portion of like bacterial infection related deaths. So scientists are currently looking at a solution for this. And the solution that they're looking at right now is bacteriophages, which are viruses that specifically infect bacteria. They were discovered a century ago, and they were initially used like in early phases, like therapeutically before antibiotics became widespread. But now, because antibiotic resistance is rising, phages are being reconsidered as like an alternative to therapy to deal with antibiotic resistance. But there are lots of advantages and disadvantages that are sort of like defining how well they can be used in clinical therapy right now. So for example, like with the use of CRISPR and gene editing technology, it can be engineered to target specific AMR bacteria. It can also be used for accurate and specific targeting of bacteria. So that way, like the microbiomes in the human body can stay stable without like wiping out the whole population of the bacteria. But there are other disadvantages. So for instance, bacteria could also like evolve to be resistant to the phages. The safety of the phages is somewhat uncertain right now. And bacteriophages could also help spread ARGs, which are antibiotic resistance genes, through the process of transduction. So based on all of this, there's currently like some uncertainties about whether or not these could be used well in clinical settings to target antimicrobial resistance. So these are just the aims of my literature review. I'll just like briefly summarize. I'm just going to be like summing up the history of bacterial infections, antibiotic resistance, bacteriophage therapy, and then the mechanisms behind all these principles, and then like the advantages and disadvantages of phage therapy, the current evidence on human clinical applications, current attitudes towards phage therapy, and how this all sort of impacts the future research that should be conducted on phage therapy and like clinical implementation for the future. And this is my methodology. I was mostly sourcing my studies from reputable sources such as like PubMed, Science Direct, or the ASM. I was using search terms like this about phages or antibiotic resistance and bacterial infection. But for the clinical trials and case report portion specifically, where I was looking at the human clinical applications, I had like more specific exclusion criteria. So some of them are listed on the screen. So that was how I was doing my search for the studies that I was using. So my first section is just about bacterial infection and antibiotics, just explaining like how it developed, its historical significance. So bacteria, of course, cause a plethora of human diseases like pneumonia, meningitis, tuberculosis, and UTI, sorry. And these are just some examples of major plagues. So for example, there's the bubonic plague, which is caused by Yersinia pestis, cholera, meningitis, and UTIs. And clearly, like as you can see from the statistics, they all have like large impacts on humanity, on different civilizations. And in response to these, in the early 20th century, antibiotics were developed. So these are the first like three main antibiotics that were developed. There was salvarsan, which was developed by Paul Ehrlich in the 1909, then penicillin by Alexander Fleming, and this was later purified and turned into one of the first antibiotic drugs. And then also Prontosil, which was created by Gerhard Domack. And these revolutionized medicine in the 20th century, so much so that it was actually dubbed the antibiotic era. And this also like allowed physicians to treat life-threatening infections, but then also perform more like important procedures. So for example, like invasive surgery or transplants, they rely on the use of antibiotics. But then after this, obviously antibiotic resistance rose up. And this was because, so the first case of antibiotic resistance, I believe that was like a big deal was with Staphylococcus aureus. This led to MRSA, which was in the UK as early as 1942. So this sort of put an end to the golden era of antibiotic discovery. And this develops because over time, bacteria will sort of naturally like acquire certain mutations which allow them to resist the mechanisms of antibiotics. And as a result of these, the resistant bacteria, as you can see in the graphic, the resistant bacteria will be the ones that survive and then they proliferate on. So then as the infection progresses, it'll develop antibiotic resistance and then antibiotics will no longer be effective against that infection. And then there's really no way to treat it if it progresses too far. So this is the current situation right now. The CDC is estimating that over 2.8 million antibiotic resistant infections occur each year, with around 35,000 deaths. And these are some of the causes, like the main contributors to this issue. So there's overprescription and misuse. So if antibiotics aren't properly administered, then it can lead to antibiotic resistance. And then also the agricultural uses, where it's often administered to animals. And also poor sanitation can just increase the spread of disease. So these are sort of what contribute to antibiotic resistance. And then bacteriophages are, of course, the solution that scientists are looking at as a solution for this problem. So this is just a brief history of how bacteriophages were first developed. So it was discovered independently by both Frederick Twort and Felix de Herel. But Felix de Herel was the one who classified it properly, and he was also the one who conducted the first clinical trial involving bacteriophages, where he used them to treat a young boy with dysentery. And although this did work out, this trial did like prove useful, and the boy was cured. And early phage therapy was like used widely in Eastern Europe and in the Soviet Union in the 1920s and 1930s. It eventually declined because of antibiotics rising, and eventually antibiotics like dominated Western medicine more so than phage therapy. But in the end, of course, antibiotic resistance then developed, and now scientists are looking at bacteriophages again. So now it's starting to reemerge in the current day. This is the general structure of a phage. So as you can see, there's the head area, which is composed of a capsid with the DNA material inside, or the genetic material inside. And then the tail portion is used by the bacteriophage to inject its genetic material into the bacterial cell. So the process of replication, there's two different processes. The first one is the lytic cycle, which is where essentially the bacteria hijacks the host cell's machinery like immediately, and it immediately starts producing new phage particles. And once enough new phages are produced, they then lyse the cell, and then they go on to infect other cells. But then there's also the lysogenic cycle, where the phage DNA integrates itself into the bacterial genetic material, and then it just sort of like lives there and survives for generation. And then eventually, due to like an external trigger, then it'll like later on go back into the lytic cycle. So these are the two main pathways, but usually bacteriophages that are used in phage therapy rely on the lytic cycle. So these are the mechanisms of phage therapy. The first step is always selection. So for a particular infection, based on the type of bacteria, a specific kind of bacteriophage will be chosen. Then this will be administered, the phage, whichever one was chosen, and then the phages that were administered will replicate within the cell. And then after this, they'll lyse the cell, and then this will keep going on until the entire infection is cleared up. So there are some advantages to phage therapy over traditional antibiotics. The main one is that because they're so specific, you can see in the diagram over here that the way that they work is you have to attach to specific receptors in the cell surface. So as a result, they're very specific on the bacteria that they target. And as a result of this, you can preserve the rest of the microbiome. So for example, the gut microbiome, though it'll preserve like the existing bacteria, and it'll only target the infectious bacteria. And it can also target antibiotic resistance because they can be engineered using CRISPR and tools like that in order to target specifically the resistant strain of bacteria. They can also self-replicate and autodose. So essentially what happens here is that they self-replicate inside the host until the entire infection is cleared. And then once all the bacteria are eliminated, the phages are essentially, they don't have anything else to infect. So they're just naturally cleared from the body after the infection is cleared up. They're also very good at targeting biofilms, which are they're very like persistent communities of bacteria. They usually develop on like inside of catheters or prosthetic joints. They're very hard to target with regular antibiotics because horizontal gene transfer happens a lot inside of these biofilms, but phages have been shown to effectively penetrate these biofilms. And then they're also good at overcoming resistance because they can sort of like co-evolve with the bacteria. So if the bacteria evolve to resist the phages can also evolve to resist the bacteria. And they're also good with combined therapy because they can work synergistically with antibiotics. But there are also a lot of disadvantages to phage therapy. So for example, there's the diagnostic challenge where usually you have to like select a specific phage in order to match that specific bacteria. And in order to do this, you need a vast library of phages. So that way you can like select a good sample. So that's a big challenge. There's also regulatory hurdles because usually like a lot of the ways that the government like monitors drugs is that they like those like standards aren't really compatible with phages because the phage therapy is very like personalized to each individual patient based on like that infection. So it's difficult to like get approval for a standardized product. And also again, with bacterial resistance, it like needs continuous research and development. This can be a big financial burden. And there's also a lot of pharmaceutical unknowns because people don't really know how it's going to interact with the body's immune system or how specifically it's going to act at the site of infection. So those are a lot of major problems. And then lastly, there is a potential for adverse reactions where when they lyse the bacterial cells, it could release endotoxins into the system, which could just like worsen the infection or cause like inflammatory responses and things like that. So those are a lot of the disadvantages. And then here I was covering the human clinical applications based on what I saw in like different studies and trials. So this is sort of a summary of like the trials I chose. I ended up choosing 10 articles that were suitable for review. 8 of these were clinical trials and 2 were case reports. So overall for the patient demographics, I'll sort of speed through this part, but they were often used as like in like compassionate use cases where regular antibiotics weren't exactly working or they were involving like chronic conditions that were more difficult to treat. So that's the major trend. Phage therapies are usually used for compassionate use conditions. For safety and adverse events, they were genuinely they were usually uncommon. These are some examples of what was going on. In some studies, there were no adverse events reported, and in one of them, I believe like neutralizing antibiotics were detected, but it didn't really like lose like the benefit of the treatment. So this was how adverse events were looking. And then for clinical outcomes, it was sort of mixed where most of them were favorable, but then with like reduced bacterial counts and like minimal relapse rates. But then some of these did show that the phage therapy was either like equivalent to the placebo or it was sometimes worse than the placebo. So this is sort of how the results were looking for that. And then for types of treatment, these are just examples of the different types of phages used. So some were commercially available phages like pyophage or microgen. So these were like pre-made phage cocktails. And then there were also engineered phages, which were specifically, or customized cocktails, which are also like specifically made for that infection. And then in this one study, they initially started with a single phage, but then because it developed phage resistance, they switched over to a phage cocktail. These are the different routes of administration. And the main takeaway from this is that most of the trials were actually using topical treatments. And one possibility for this could be because topical treatments like claim to like decrease adverse effects, even though like across the studies that I was reviewing, adverse effects were usually like comparable, and the topical treatment didn't seem to have any effect on that. And then here were the dosages and concentrations. So it was very varied, the kinds of dosages that were given out. And this also becomes a problem later on when like analyzing different trials. Oh wait, did I run out of time already? Or
Okay, just continue.
okay, sure. Okay. So yeah, the other big thing was that most of them were being used in combination with antibiotics. So here are the different examples of that. And these were usually to like try to prevent increased resistance or like try to like activate like phage antibiotic synergy. Like I explained before, they can usually work together to increase their effect. And then here, only one trial actually developed phage resistance, which is where the phage obviously like it develops resistance to the phage cocktail. But again, once they switched to like this was because they initially only administered one single phage. Once they switched to like the cocktail approach, which combines multiple phages, then this reduced resistance. Okay, so this is current attitudes. I'll go through this pretty fast, but the main thing right now is that there's growing interest in optimism, which is because there's being more research conducted. Like I mentioned, most of the trials are done for compassionate use cases, and these are like proving to like turn out well. So it's increasing confidence. And then patient and providers, they don't really like, there's not much awareness right now, but they are like willing to try phage therapy. So they don't really know like details about phage therapy, but like there's like positive attitudes toward it. And then again here, there's the issue of data access pops up again, where like people need to have access to large banks of phages in order to like make the therapy well. And then there's also regulatory hurdles, like I mentioned, with getting approval for these products. And then finally, there's the limited awareness issue of it again, where most providers don't even know that this exists. So even if they're willing to try it, they aren't aware that phage therapy is an option that they can use. Okay, so for my discussion section, in general, phage therapy is growing as an alternative to antibiotics, and while it does have lots of advantages, it also does have disadvantages that need to be accounted for. So, although there are promising results, consistency is like very much needed across the like different trials in order to like determine like a standard procedure for how to conduct like larger scale trials and how to like assess data between trials. And in particular, the disadvantages is like concern like the feasibility of making it applicable to patients and like getting it through like regulations. And then these are just different avenues for future development. So obviously just like more research into phages, antibiotics, how they impact ARG dynamics, and then also just standardization, again, like I mentioned, is the biggest thing across trials. For my limitations, the main thing again was that It's just a lack of standardization among trials. Like it makes it difficult to analyze usually. And then for the conclusion, this is just a summary of everything I mentioned. They are an emerging alternative, but there are still lots of challenges that need to be dealt with in order to make them more feasible. For my acknowledgements, I'd just like to thank Professor K, Professor Prahlad, and Professor Torimoka. Sorry if I pronounced that wrong. But just like for all of your help with like planning, editing, and revising, and like working to publish this paper. Like it would have been impossible without all of your help and support. So thank you for that. And then here are my citations. Yeah, sorry, that took very long. Sorry about that.
All right, it's okay,
Yeah.
Ananya. Let's give Ananya
Yeah.
a clap. Congratulations. Your presentation was both thoughtful and articulate, and you showed not just strong research skills, but also a genuine passion for your topic. All right, now let's hear from our experts. We'll start with Professor Prahlad.
Hey, no, that was a great talk. Yeah, it was a little too long, but you learn. Okay. Yeah.
Yeah, I thought it
But
would go by quicker. Sorry.
yeah. Let's have a quick question. So is there any FDA approved therapy for like any of the resistant bacteria using phage?
I think like very early on in the 1900s, there was one company that developed one. It was called like Eli Lilly, but I don't think any modern ones have been made, but there are lots in like Eastern Europe, I think.
Okay. Okay. Yeah, that's a great presentation once again.
Yeah, thank you.
Congratulations. Yeah.
Thank you, Professor Prahlad. Professor K, would you like to say something? Oh, sorry. I think you're on mute.
Oh, hi. I hope I will not be cut off again. Okay, but Ananya, right? This is not your first hurdle in research, although it's your first time to do conducting independent research. How did you find your experience in conducting independent research with Gifted Gabber?
I think it was really helpful because like I think you guys like provided a really good framework to like understand what the process is like and like the steps we should take. So that was really helpful. And then also like just I think the research itself was like really informative and it definitely like helped me like grow my skills and like learn how to do these sorts of things better. Oh, I think she might be frozen.
Oh, sorry. Okay. Yeah, I think Professor K is having a problem with her connection. Well, thank so much, Professor K. Uh, Professor Pilar. So continue. Now we'll proceed to the next presenter. We have, of course, Salar.
Yeah, hello. Okay, I'll share my screen.
All right.
Sorry, one sec. Okay. So, hello, my name is Salar Fez. I go to Van Tantrup High School. This is my thesis defense presentation for bispecific antibodies in cancer treatment. This is a literature review article. So let's get started. So for decades, cancer treatment meant surgery, radiation, and chemotherapy. But now we have this new innovative thing called immunotherapy, which gives power to the body's own immune system and use of the body's own immune cells to fight cancer. Monoclonal antibodies are a recent innovation to immunotherapy, and it targets one antigen in the body, which does have its own limitations in treating cancer, which will be discussed later. And bispecific antibodies are a new method of fighting cancer, and it's a combination of two different monoclonal antibodies. In this article, in this presentation, sorry, I'll be talking about the design, clinical applications, advantages, challenges, and the future research being conducted on bispecific antibodies. So for the methodology, the following websites were used to find articles and studies. I used PubMed, SciSpace, clinicaltrials.gov, Scopus, and Google Scholar. The articles were from 2000 and above and in the English language to keep them relevant. And the search terms were bispecific antibodies and cancer. And I used scientific diagrams that were made in BioRender. And I used the APA format for citations. So before diving deep into the subject, I want to talk about the basic structure of antibodies so we can make a base for future understanding. So we have a heavy chain and a light chain, and you can see the lower and central part of the antibody is labeled as the Fc region, and this is what interacts with immune cells and complement proteins. The other part of the antibody is the Fab region, where it recognizes specific antigens and binds with them. In the Fab region, you can see the top two parts of like the Y. This is called the variable region, and this is the part that actually does bind with the antigen. And this is the antibody in your like own, I guess, like body. These are natural, and they only target one antigen across both of the Fab regions. So there's two types of bispecific antibodies. So now that we understand what an antibody is, I want to go deep into ABS, and for IgG-like bispecific antibodies, we have two different monoclonal antibodies, and we take half of one and half of the other, and we combine it to create one antibody that targets two different antigens. So we take the Fab region of one different monoclonal antibody and one different monoclonal antibody and combine them, and we keep the Fc region. And in this diagram, you can see there's the CD20 antigen like receptor area. We take that, which that's like CD20 is like an antigen on tumors and cancer cells, and we have the CD3 antigen like binding site, which is on T cells, which like kills cancer. So then we're bringing a tumor cell, a cancer cell, and we're putting it next to a T cell, which allows the T cell to destroy the cancer cell. For non-IgG like antibodies, we have a bunch of more variation. So I'll start off with the BITE. And for the BITE, we take only the variable region of antibodies, and we get rid of totally with the Fc region, and we only take the variable part. So these are a lot stronger and can penetrate more, but since they're so small, they become a lot less stable, and they can break apart much easier than IgG-like antibodies. So to deal with the instability of this, we move on to C, the SCFVFC, where we only have the variable regions, but we do keep the FC part. So where we have half of the Fab in the full FC version, which does create a nice balance. But to further balance this, we go to D, the mini body. In the mini body, we have half of the Fab and half of the FC region to create like a super balanced, like small but effective antibody. And E is the nanobody, which is something I think is pretty special. It comes from camels, and for some reason in camels, they only have the heavy chain, and it's a normal antibody, but it functions at the same rate with only one out of the two chains, which makes them much smaller and potentially more effective, or hopefully more effective. So moving on to like the more results part, different studies here on hematologic malignancies, and they have bispecific antibodies have a lot of success, especially in multiple myeloma, and they have FDA approval for all these like four cancers I've listed that are on this slide below. But there has been less success with solid tumors due to physical barriers around the solid tumor. Obviously, it's like solid, and there's a lack of specific antigens on solid tumors. There's a lot of tumor resistance. However, for the 5 cancers on this slide, there has been breakthroughs with some bispecific antibodies, and they work on different growth factor receptors on solid tumors to stunt their growth, and they do also engage T cells with tumors. So moving on to like the more discussion part, they have advantages. So to begin with, bispecific antibodies can physically link tumor cells with T cells, which bypasses the need for direct recognition and overcomes immune evasion. Our next advantage is that bispecific antibodies can block 2 signaling receptors at once, which prevents tumor escape and resistance mechanisms that often undermine single target therapies. Our next advantage is that bispecific antibodies can be engineered into smaller fragments for more tumor penetration, which obviously does have that issue with them being less stable. And bispecific antibodies also reduce the off-tumor toxicity and spare more healthy cells. Moving on to the challenges, there are some manufacturing hurdles with complex engineering required to ensure like the correct chain pairing. And there are some pharmacokinetics, kinetics and biodistribution complexities with the IgG-like antibodies being bigger and having issues with penetrating the tumors, but non-IgG-like antibodies are too small and they're unstable. And another challenge is cytokine release syndrome or CRS. And basically, what happens is when you put the bispecific antibody into the cell, it causes widespread immune activation where your immune system is going on or the part of the body that has the cancer and all around, which causes the deaths of some healthy cells. And bispecific antibodies require like specialized manufacturing platforms, and there's a bunch of stringent regulatory requirements for bispecific antibodies, which makes research and production a lot more costly. Our future innovations, so the first future innovation is like the next generation engineering, which includes multiphasic and triphasic antibodies. So you know how there's the bispecific antibodies, we have two different like antigen binding sites, but for multiphasic and triphasic, we can have three or more, which make them a much more efficient. And our next future innovation is improved safety tools, which like would include like an off switch, which if there's like sorry, if it includes a novel designs to minimize cytokine release syndrome. And our next innovation is there's development against new tumor-specific antigens and new fragment-based bispecific antibodies to target solid tumors. And lastly, there has been integrations with like common treatments like chemotherapy and radiotherapy, and like combining that with bispecific antibodies to make them more effective. Going on to the conclusions. So the bispecific antibodies are revolutionary therapy and they're transforming oncology with several FDA approvals for hematologic cancer and many more coming in development. And there's expanding applications for bispecific antibodies. So while solid tumor approvals remain limited, a growing pipeline holds promise for future and broader uses. And a lot of future potential with advances in design, safety, and integration of other therapies. And this might make bispecific antibodies to go from being a cancer care to a cancer cure. So I want to give special thanks to Professor Prahlad Parajuli and Professor K. Diaz and Professor Torimoka for helping me out with this process. I definitely would not have been able to do it without them. I feel like they gave a lot of information for everything. So I want to give a special thanks. And that's all. Thank you.
Thank you so much, Salar. All right, you demonstrated impressive clarity and confidence in your delivery, and the way you structured your argument shows really maturity in your thinking. All right, now let's give the floor to our panel of experts for the comments and suggestions. We'll start first with Professor Prahlad.
Hey, that's a great presentation, Salah. Yeah, good job there. Congratulations. And so, I have a so the multiphasic antibody. Did you find any example of any multiphasic antibody against any cancer in the clinical trials?
Well, say I actually did not. I feel like it's still very, very new. Like I don't think they've done the clinical trials with them yet. They're still developing them, but hopefully in the future we can have them.
So, what kind of combination would help with other therapies? Do you have any comment on that? So, because you keep saying that the antibodies, because they don't penetrate the cancer, but they have to drag even if you have like make a small molecule with only the Fab, they have to drag the T cells or NK cells with them, right? So that needs a bigger space, so it doesn't matter. So what kind of combination would help? Did you find any idea how to make it possible so that they can drag the immune cells also with them?
So I think it's a very difficult topic because we have to find the perfect balance kind of between like size and effectiveness. So
Mhm.
did think like the mini body that I showed with like half of the fab and half of the FC would be a really nice balance, but I couldn't find like that many like clinical applications applied yet. So maybe they're like that's still like in progress. But I feel like that, in my opinion, was the most like effective one. That would be the most effective one.
Mhm. Probably you want to mention something about immunogenic chemotherapy. Okay, so with chemotherapy, so that actually kills the tumor cells a little bit, that opens the space, and then they can also generate some immune response so that that the T cells and other cells can enter. Okay, so maybe
Yeah,
mention that in your
that sounds
yeah.
super interesting.
Okay, awesome job. Congratulations.
Thank you.
Thank you. Thank you, Professor. Professor K.
Hey, hi, I'm Beth. Okay. All right. I believe, Salar, you had a lot of difficulties when you were conducting your research, but what would you say was the most challenging part of this entire process for you?
I think the most challenging part is kind of just like I don't know, I feel like taking the words you're seeing from articles and like taking all this like super complex stuff and just putting it into words to make them like be able to be understood by a lot of people, and I feel like that was really difficult for me.
Okay, thank you. How was your experience so far doing independent research?
Um, I feel I really liked it. I feel like it's a really nice challenge. I really like the experience Gifted Gabber has with like you're just learning stuff all the time, but it's not like you're just gonna write it for us. Like we have to. It's still not just like a. It's super independent. I feel like I kind of like that aspect of it.
Thank you. You did a good job, by the way.
Thank you, Professor K. And thank you, Salar, for that great presentation. Okay, we'll move next to our third presenter. We have Varsha. Varsha?
Hello.
Hi, dear.
Sorry, one second. Okay. All right, could I get started then?
Yeah, sure. You go ahead.
So, hi, my name is Varsha Murali, and my project was on environmentally induced endocrine disrupting chemicals, EDC's effect on increased gender identification discrepancies. So just a little bit about myself. Yeah, my name is Varsha. I go to Taft Charter High School in Los Angeles, and my research purpose was to establish the link between environmental factors and increased gender transitions, to increase knowledge on gender transitions to improve trans healthcare, and to establish a biological basis for gender identity. So, my introduction. Gender identification disparity is a difference in one's gender identification versus birth sex due to hormonal or physiological abnormalities, which are often linked to environmental pollutants. The environmental pollutants that are linked to hormone physiological dysregulation are called endocrine disrupting chemicals, which, for the purpose of this presentation going forward, we're going to call it EDCs. Your endocrine system is a network of glands and hormones that produce, secrete, and regulate hormones. This system works with others to ensure one's development and function. Endocrine disrupting chemicals, EDCs, however, are substances that cause your endocrine system to not function properly, leading to a plethora of problems. This study examines how environmentally induced EDCs affect increased gender identification discrepancies. So my methodology, the research type for this paper is a secondary literature review. This is a comprehensive summary and analysis regarding EDCs, gender identification discrepancies, gender identity, and sex ratios with a focus on clinical studies. After synthesizing information from the literature, this paper identifies gaps and areas for potential future research. Literature selection criteria were literature related to the keywords on the slide, and only peer-reviewed journal and tier 1 sources were utilized. Due to potential changes in the environment, only sources written in the last 10 years were used in the literature. So here's just an example of two really common EDCs, so we can really understand how pervasive this is to daily life. So the first example would be bisphenol BSP, which is a man-made compound that is commonly found in plastics and food packaging. If we're looking at the image on the left, those are things that we would use literally every day if we're going out to eat, if we're taking food to go to school or work. And then phthalates are plasticizers in production that disrupt the androgen signaling. Again, plastic water bottles, just really pervasive to daily life. One of the endocrine system's major processes is the development of the reproductive system. Sex determination occurs early in gestation and is controlled by a very minimal amount of hormones, such as luteinizing hormones, LH, FSH, and testosterone. EDCs can mimic these hormones and estrogen function and interfere with male-female sexual maturity and hormones. However, the most concerning aspect of EDCs is that they can even cross the placenta, affecting fetuses in utero, thus affecting the reproductive cycle development. Thus, exposure to EDCs disrupt these hormones, lead to disruptions in sexual development, and cause additional diseases. In cases of high exposure, this often leads to gender identification disparity. So, the endocrine system or hormonal system is involved heavily in pregnancy, such as implantation, placental formation, maternal adaptation, embryonic and fetal development, birth, and the fetus's adaptation to post-birth life. Both genders' fetuses produce the hormone testosterone. Testosterone is crucial in fetal sexual differentiation, and exposure to high levels of testosterone is linked with polycystic ovary syndrome, or PCOS. The aromatase enzyme in the placenta converts androgen into testosterone, which protects the female fetus from maternal androgen. Many polymorphisms of aromatase have been discovered and linked to sex hormones and hyperandrogenism. Other important hormones in fetal development are estrogen, progesterone, and estradiol. Estrogen and progesterone are produced by the placenta after implantation. Estrogen affects fetal vasculature by acting on endothelium and vascular smooth muscle through rapid signaling pathways and genomic mechanisms. Estradiol is another sex hormone that fetuses are exposed to through their mother, gonads, and synthesis locally in the brain. Estradiol is involved in sex differentiation. These sex hormones all impact sexual differentiation and identity. EDCs in the environment, particularly estrogen mimetics, disrupt normal brain development and sexual differentiation. So connecting all this information back to the two examples of EDCs that we discussed earlier. So BSP, first of all, that we're looking at here is a man-made compound commonly found in plastics and food packaging with the ability to mimic estrogen and bind to estrogen receptors. This activates unnatural gene transcription. BSP exposure during fetal development and early childhood can lead to issues in reproductive tract development, puberty timing, and neural pathways. Furthermore, BSP is considered an EDC due to its ability to act like xenoestrogen. This provides BSP with the ability to interfere in many estrogen-related pathways, causing hormonal diseases, sexual behavioral problems, and accelerated puberty, showing this EDC's strong link with gender identification discrepancies. Next, phthalates are used as plasticizers in production disruption and they disrupt androgen signaling. They disrupt fetal Leydig cell development. So these cells are crucial for testosterone production and other hormones necessary for the male reproductive tract formation. Exposure to phthalates reduce steroidogenesis and gene expression in the fetal testis, leading to reduced androgen. These disruptions can lead to those fetuses being born with male reproductive tract anomalies such as cryptorchidism, which is an undescended testis, and hypospadias, which is abnormal genital opening. So the reason this is so significant to gender identity is that gender identity is based on brain development. Brain development is shaped in large part by fetal hormonal exposure and levels. So sexual dimorphism is largely attributed to the influence of testosterone, estrogen, and other sex hormones during fetal life. Studies pertaining to transgender individuals have found that certain brain features correlate more with gender identity than sex assigned at birth, proving the biological basis for gender identity. So, in conclusion, studies provide strong evidence for a causal relationship between EDC's hormone levels, alterations, and gender identity. This provides evidence for the biological basis of gender identification. Through understanding the role certain hormones play in fetal sexual development, as well as how EDCs inhibit those processes, the need to reduce EDCs is well outlined. So, some limitations. So, we were all told to kind of use clinical studies, but the problem with mine is Professor Pahad and I spent a long, long time poring over clinical studies that could be used, but the research that is specifically linked in EDCs to gender identification, we weren't able to find any. There was only studies that were finding the link between like reproductive outcomes or reproductive tract abnormalities. So there was one that was called Lead Endocrine Disruption and Reproductive Outcomes, and was a study sponsored by the National Institute of Environmental Health that studied lead's effect on the endocrine system and reproductive outcomes. And this other one that studied hypospadias, it's a birth defect affecting the external genital organs of boys. But yes, none of these were actually related to gender identification, which was definitely a limitation of my study, is that there wasn't really any on them. So because of that, I propose that future work on this in this area be focused on case studies that strengthen the causal relationship between EDC's hormone level alterations and gender identifications. In terms of mitigating the effects of this issue, the research should focus on also how to tailor medical care for trans individuals, as well as the effect of this issue. To tackle the core of this issue, though, alternatives for EDC should be found, especially in plastic packaging, but also cosmetic children toys. We do currently have regulations in the US for EDCs, but due to plastic companies lobbying, they often aren't enforced. So that's definitely something in the more political science arena for looking cross-disciplinary that should be looked into. So thank you, everyone, and thank you to Dr. Prahlad, who just very patiently sat with me looking through all the clinical studies and was just super helpful in this process on how to structure this whole presentation. Professor K, who wrote out an outline for every single one of us on what to write about in our papers. That was really, really appreciated. Thank you. Professor Torimocha, who helped us on how to structure this presentation and I think made us all a little less nervous for talking today. So thank you all of you guys for your support. I really appreciated it, and I'm ready for questions.
Thank you, Varsha. Your work stood out for its depth and, of course, thoroughness. So congratulations, job well done. All right. Now let's hear the comments and suggestions of our professors. We'll start.
Hey, great work there, Versa. Congratulations. Uh, one quick question. There are some like genetic predisposition, also some genes also involved in, gender determination and identification, right? So is there any relation, some of the genes, does it make some individuals more susceptible to those like chemicals that you mentioned? Did you find any evidence to that?
None of the work that I looked on did find anything to that, and I think that's in part due to there isn't really any studies looking at people specifically in this. A lot of what I used was background information on what EDCs are and potentially how they could affect, but due to the lack of case studies, I didn't come across any information on that specifically.
Okay, good job, Ken. Congratulations.
Thank you.
Thank you, Professor Prahlad. Professor K.
Okay, hi, Varsha. You were among those who were really passionate about your paper from the very beginning, but for some reason, because of like now I understood, there was a big gap on the submissions of your paper because you had difficulty in sourcing for clinical trials. So how did you handle all the setbacks or the difficulties in the process of doing your research? When you were like from the very beginning, you were very keen on submitting on time, and then there's this lull. How did you handle all that?
I think I found a lot of information in the beginning on like the background of the issue, and I was like, great, I understand how this works. But then I couldn't really find anything that specifically connected gender identification to these hormones. So after a couple of weeks of poring over clinical studies and not finding that, I kind of looked at more like, okay, I looked at like the hormonal aspect of it, and like then I created that link myself to how those hormones are going to identify gender identification. So it's more of like a two-step process where I had to connect A to C rather than A to B. So that's kind of how I tackled that.
How has your knowledge of this topic changed from the beginning to now?
Um, I think one of the major things that I found really interesting with my topic was how brain development is impacted, especially how trans individuals have characteristics of their gender identity and not just their sex assigned at birth. And I think that really changed my understanding of gender identification, but also made me more passionate about like another tangent of this topic, like trans healthcare. But just learning like the biological basis for gender identification was just really surprising to me, but I also really enjoyed learning about it.
I'm enjoying, amazed also at the same time. I'm learning about it today. So thank you for that. Congratulations.
Thank you.
Thank you, Professor K. Let's give again Varsha a virtual clap. All right. Job well done, Varsha. Okay, don't forget again to send in your questions in the chat box after each presentation. All right. Now we'll proceed to our fourth presenter. We have Amarish.
Oh, hi there. I'll just share my screen real quick.
All right, sure.
So, hi, hello there. I'm Omarish, and I am an 11th grader at West Ranch High School. And today I'll be going over my presentation on my comparison of vaccine platforms, which is a scholarly review of mRNA, viral vector, and traditional vaccines. So my table of contents include an introduction, followed by a literature review, followed by the methodology, as well as how the vaccine works in the immunologic concept. And uh, preceding that would be my findings, discussion plus tables, my conclusion plus recommendations, and finally my study limitations. So in terms of the introduction, vaccines were pioneered by Edward Jenner, who's the father of immunology, and they've been one of the greatest achievements in medicine ever since, preventing millions of deaths per year. And so my focus was during COVID-19 and a bit after and before, where three main vaccine platforms, traditional, viral vector, and mRNA, became the foundation of the global responses. Now, vaccines are currently being explored for cancer therapies due to the advancements in the field of mRNA. And the research question that I kind of came up with and considered would be, how do these vaccine platforms compare in science, safety, and global accessibility, with a strong eye on efficacy? So in terms of literature review, the question that I was sort of keen to answer would be, what do we already know about vaccines and their gaps? So traditional vaccines have been used for quite a while, and they're proven safe over loads and loads of tests. However, they are very slow to produce during times where the disease is rapidly evolving, such as epidemics and pandemics. For viral vector vaccines, they're also very effective but widely accessible, but the ability for clotting events due to the increased production of platelets to occur reduced its trust among among consumers. And so for mRNA vaccines, they're quite recent, they're well versed in speed and efficacy, but the reason for their distribution being limited is because they have to be stored in a very, very cold environment. And so because of this constraint, it is not as well distributed as the other two vaccine platforms, sorry. And so in terms of the gaps, there are a few comparative reviews that exist for mRNA vaccines, specifically as they're being pioneered and they're a relatively new phenomenon. And also the field of cancer applications is still emerging simultaneously as we speak. So in terms of methodology, I reviewed through approximately 145 peer-reviewed studies published between 2019 and 2025. I did exclude quite a large proportion of my studies due to either they had a poor methodology, a small sample size that wasn't simply just representative of the population, or a lack of empirical data, as it wasn't what I was trying to go for. So my final analysis incorporated 12 references that is subject to change depending on corrections in my research article and to what I believe to be the most effective of articles. And so the reliable websites that I used include clinicaltrials.gov and PubMed, with an approach that consists of my goal in identifying patterns in safety, efficacy, accessibility, and cancer applications. So what is the science behind vaccines? Well, vaccines introduce antigens, which are processed by dendritic cells, essentially in the neurons, and they're essentially identified by memory cells, so that in future altercations, the same disease is already familiar to your immune system, and so it will not be as effective as it was first. And it's also presented on MHC molecules, which act as a billboard for the cell to let the surroundings know what's happening inside of a particular cell. And so what happens after this? Well, CD4 plus helper T cells, which are the cells responsible for formulating immune response, as well as the cytotoxic CD8 plus cells, which partake in the apoptosis of abnormal cells, do their assigned jobs, as well as B cells, which produce antibodies, and as you said, memory cells, which ensures that the disease won't harm the immune system in the long term. And so in terms of cancer vaccines now, they apply the same principle as what I've spoken about previously, by targeting neoantigens, which is a specific type of antigen like HER2 and ROS. And they do this by helping the immune system attack the tumors, in short, and prevent the uncontrolled cell growth of tumor cells. And yeah, there's a diagram below, which was all credits go to genome.gov. And essentially what's happening here is this is specific to the COVID-19 vaccine. And so what happens is you have a spike protein being introduced by the vaccine. And so once the ribosome assembles all the components needed to formulate an amino acid change, sorry, the spike protein is induced by antibodies and other immune cells, so that it can be so if that in future altercations, like I said, if the same disease tries to attack the immune system, it will be efficiently dealt with, as the diagram shows. So what did I find in all my vaccine, amongst all vaccine types? So my insights, just going over them since they summed up what I was trying to get at, was that mRNA vaccines were the most effective, but obviously, as I said before, they're the hardest to distribute fairly. Viral vector vaccines do fill the gap where low resource regions might not have access to as many items as a well-developed region, but it does still face trust issues in terms of clotting or rare clotting occurring when someone takes the vaccine. And traditional vaccines still do remain the foundation of immunization because like I said, they're very well known and people have a strong trust in them. But obviously, they do lack speed in urgent cases where a certain population needs vaccines to fight off diseases. And now in the field of oncology, mRNA vaccines are leading personalized cancer vaccine developments because their speed and efficacy is like unmatched in comparison to the other two platforms. And once developed fully, they can be used instead as opposed to the other two types of vaccines. And here are some pros and cons related to all the three types of vaccines. And obviously, as you can see, AstraZeneca, Pfizer, a main focus on COVID-19 vaccines that many of you have taken during COVID. And I've made two tables that go over the type of platform and how effective it is with the pros and cons. And in the second table specifically, I've gone over the vaccine platforms in terms of cancer specifically. And so in terms of my 4 points, vaccine innovation, I think there is an element of science to it, but it's also shaped by logistics, equity, and public trust. And for mRNA vaccines, despite being fast and very effective, it also did reveal inequities in infrastructure worldwide when I was researching through that. And for viral vector vaccines, it did show um, a, a sort of compromise, if that makes sense, because less potent but essential, they were less potent but essential where storage was impossible. And so kind of tying up with traditional vaccines in the summary. So traditional vaccines, like I said before, they get the job done and they're trusted by many of us over a long period of time. And I think together these platforms complement each other with their pros and cons and ensure resilience in global health. So in conclusion, like I said in the last slide, no single platform is better than the other, where I think like each platform complements each other based on their strengths and weaknesses. And now in terms of future vaccinology, uh, it should involve innovation, equity, and collaboration to meet global needs, such as when COVID hit us immediately towards the end of 2019. And so in terms of mRNA, viral vector, and traditional, mRNA was the fastest and most adaptable. Viral vector was very cost effective and practical in low resource regions. And traditional is safe and trusted and is the backbone of public health in the United States and globally. So for my recommendations, in terms of infrastructure, definitely expand cold chain systems or systems where you can preserve mRNA vaccines specifically, since in my opinion, they pose as the most effective and useful vaccine. Um, in terms of equity, fair vaccine access should be brought to all regions of all income levels. Research should definitely continue developing vaccines with a specific interest in cancer vaccines. And finally, public trust should definitely counter misinformation, such as like the MMR scandal in the early 2000s, and promote vaccine literacy worldwide, which is essentially how do vaccines work, presenting all this sort of information to the ordinary public. And so finally, my study limitations. So in terms of what I did, my study limitations would be more so the public. I had to only focus on COVID-19, not really any other specific event. And I also focused on a select amount of articles and clinical trials. So that would be the limitations on my end. But in terms of the reviews I've actually used, as well as the information I've provided, I'd say that all the studies I used were English language, so no other languages were used, which could limit like the representation of potentially crucial data. And so, on that note, in terms of geography, most of my information came from North America, Europe, and Asia. Um, so regions such as Africa and Australia didn't have as much representation. And I think in terms of bias, there is some publication bias I feel with some of my clinical trials and publications because some producers definitely or people who take part in the trials, they'll definitely keep the positive results and withhold the negative results, just to make themselves more appealing to people who do scholarly reviews. And finally, I think future research should definitely include diverse populations, a larger sample size, which is representative of a certain group, and longitudinal studies, basically over longer periods of time to to denote more accuracy in such studies. And so thank you for listening. And I really do want to credit Professors K. Diaz, Virgil Toremoca, and Paula Parajuli, because all of them have assisted me with their invaluable knowledge, where Professor Diaz helped me with structuring my research draft and obviously putting a title that's quite catchy. And um, uh, Professor Parajuli also helped just today how to restructure my research draft and also the information that I might want to include rather from different perspectives rather than just a certain narrative. And Professor Torimoka helped with information regarding how to submit your paper. So I found that really valuable as it's my first time doing so. And I'd also like to thank Coach Joe for giving me an opportunity to see the research I can accomplish during the period of her research program. So thank you very much.
Thank you so much, Amarish, for that great presentation. Your critical analysis was excellent.
Thank
Very
you.
good. You're welcome. All right, now let's hear the comments and suggestions from our panel of experts. We'll start with Professor Prahlad.
Hey, Amaris, yeah, great job, great
Thank you, sir.
presentation. Congratulations. So, for the audience, like there are two kinds of vaccines. Like mostly from vaccine, we understand the prophylactic vaccines that we take for infectious diseases. But there is in cancer, it's more like therapeutic, right?
Right, yeah.
So can you shed light on that a little bit? And did you find any like therapeutic vaccine using mRNA for cancer in the clinic?
So I think if I just jog back to my table, so I think for HPV, that's a prophylactic vaccine, so like I said, infectious. But in terms of like therapeutic vaccines, so related to do with cancer, like prostate cancer, I think with those vaccines, the process with delivering those vaccines is actually significantly different. So for those vaccines, they're wrapped in like a lipid like nanoparticle, and then what happens is they're transported within the cell membrane and they're reassembled to produce an antigen, which induces a response from the immune system. And so what happens is, I think with therapeutic vaccines, especially with um, like various types of cancers, my hope is that similarly to prophylactic vaccines being already developed in terms of traditional in terms of the realm of traditional vaccines, my hope is that therapeutic vaccines will be developed likewise and treat more types of drugs. But yeah, I'll make sure to research that because that's actually a good question. So yeah, thank you.
Excellent. And just for you to remember, just like in prophylactic vaccines, in cancer also, the memory response is important to prevent relapse of cancer. So
Okay.
it works as both therapeutic and prophylactic in that sense. Okay.
Okay, both ways. Okay.
Okay. All right. Good job.
Thank you,
Congratulations.
sir.
Thank you, Professor Prahlad. Let's hear Professor K's comments.
Okay. Hi, Amaresh. For someone who is very new in research, you have grown so much. This is your first hurdle.
Yeah.
Right? You didn't sound like one. Okay. Anyway, but to advise someone, or what advice would you give someone who's just starting their first research, when considering your experience?
So I'd say for someone starting off, you definitely, I think like you like gave like a structure for me. So I definitely say like structure your research paper in like with the titles and stuff, because I feel like when you start immediately and you kind of have nowhere to go, it's better to have like a sort of like template, like you can research elsewhere and see kind of like on other publications like how do they structure their work, and kind of depending on what you're researching, I feel like definitely start with a title, a sort of template to like kind of sprout ideas in your mind. And I'd also say it depends on like you should definitely check what journal you're publishing it to, because for I think in our case we had ACS formatting, whereas in other journals they might utilize APA formatting. So that's definitely something to be aware of, because I feel like most people would definitely do the wrong type of like formatting unless they've looked at the guidelines as per a publication journal's like um criteria. So yeah, that's kind of where I'd go off of.
Okay. What would you say about your overall research experience?
So in terms of research experience, I think it's definitely like not what I expected, because I've seen like just like out of curiosity, I've seen like papers online, and I've like seen how they've how they've like created like large amounts of text from like just like simple information. And my first intuition was like, oh well, I think I could do that. But then actually when it comes to actually writing a paper, it's a whole different ballgame. I think it's also about, proofreading what you've put in first, but also a way to like dissect data from another article or clinical trial and put it in your own words. I found that a bit of a challenge. So it's quite a I think rewarding experience is the way I put it. So
I'm glad that you said rewarding. I thought you would like um traumatized by this one, but congratulations, you did a really good job.
thank you.
Thank you, Professor K. Let's give Amarish again a virtual clap. Congratulations. Job well done. Okay. So I hope that all of you will continue your your writing. I know that writing and research is kind of difficult. It's challenging. Sometimes it will really exhaust you, but it's fulfilling. At the end of, the process at the end of the program, you'll be able to realize how fulfilling it is to write your own research work. So congratulations for that, Amarish. Let's proceed to the next presenter. I think Sarah is not here. We'll proceed with Sangeeta.
Yeah, hi. Give me a second. Make sure my camera up and running. Cool. Okay. Yeah, just give me a quick second.
Yeah, sure.
It's the wrong. Can you see my
Yeah.
screen? Okay,
We can.
cool.
Yeah, you can see it. Put it in
Yeah.
presentation mode. Yeah.
So, oh, so yeah. I'm Sharon Scudder. A little bit about myself. I am a junior in Interlake High School in Bellevue, Washington, and this is CRISPR-Cas9 for treating genetic blood disorders and its promises and its limits. So, genetic diseases have been plaguing humanity for thousands of years, whether that be through hereditary diseases and cancers. We haven't had a solution, but thanks to modern medicine, now we have the potential for a solution to go resolve these afflictions. Gene editing has been around longer than we can remember. Humanity has been modifying organisms for thousands of years, dating back to the Mesopotamian civilization. We've been doing things like selective breeding to go control how much wool a sheep would have or how big a chicken would be. But now, with modern medicine, we no longer have to wait thousands of hundreds of years to go get a small result. We can go do something and we can go change the genetics of a living being within a couple of years. Modern day gene editing was created in 1950 with developments such as DNA priming and base editors, which pushed this technology forward. But in recent years, a new improvement in gene editing known as CRISPR has taken the spotlight. CRISPR was created by Emmanuel Charpentier and Jennifer Doudna, and this tool was fairly recent, and it was presented as a viable option for gene editing back in 2020. And CRISPR works by using a designated guide RNA to locate a specific DNA sequence. Once at the site, the Cas9 protein then cuts both strands there. The cell repairs the break either by non-homologous end joining, which often disrupts the gene, or by homology-directed repair. But that only works if there's a DNA template that's provided. But newer tools such as base editors and prime editors can rewrite single cells or small segments without having to fully break the double strand. In medicine, there's two ways of delivering CRISPR. That's either through ex vivo, which is out of body, such as taking a small, like some blood, putting it into a separate location and editing the blood cells there, and then injecting it back into the body, or by in vivo, just in body, using carriers like lipid nanoparticles. And here's a graphic. Credit goes to reprocell.com. And so here we can see a DNA strand being edited by CRISPR-Cas9. The guiding RNA strand, it's a purple string there, and this is the DNA sequence. And these are the two end results, which are the homologous direct repair, which inserts a gene, and then non-homologous end joining, which deletes the gene. So for for my methodology, I source information from sources such as PubMed.org and Google Scholar in order to observe journals regarding CRISPR-Cas9. To look at trials regarding the use of CRISPR-Cas9, clinicaltrials.org was used to look at the effectiveness and use of CRISPR-Cas9 as a viable solution. And I sort of just used that to go compare how is it practical, what are some human trials going on, and how have they been working. And the other sources such as the Food and Drug Administration, Nature Medicine, and the New England Journal of Medicine were used in supplementing my analysis by sort of giving proof that, yeah, this works as a viable solution and the government has approved it. Yeah, and so throughout my studies, I've learned that CRISPR works best in monogenic hemoglobinopathies, like when enhancing the BCLA enhancer or HBB promoters to lift HB fatal hemoglobin levels. The results from the exa-cel treatment, which is the only like the biggest viable treatment using CRISPR-Cas9 in regards to sickle cell disease or beta thalassemia, it shows real world like gains by using transfusion independence and fewer vaso-occlusive crises. And this shows that CRISPR can deal with monogenic disorders like SED and beta thalassemia. But what about polygenic diseases like blood cancers, which are the target of today's study? In cancers, unlike the traditional methods of applying CRISPR to go attack tumors or cells gone rogue, research proves that CRISPR works best when CRISPR-Cas9 is used to engineer the other cells to fight tumors better. For example, in acute lymphoblastic leukemia, CRISPR is used to engineer the CAR T cells to improve the proliferation and effect on immature lymphoblasts rather than directly attacking the tumors itself. And in these blood diseases, there's way too many genes to go edit, and it's not currently viable with the CRISPR technology we have right now to go and edit all of these genes to go get rid of these tumors. But by engineering certain immune cells, which only require two genes to activate, we can use them to go fight tumors more effectively. Here are some studies that I've gained from clinicaltrials.org that show the use of CRISPR against diseases such as transfusion development B thalassemia or sickle cell disease. You can see what exactly the treatment was and the results here. Yeah, thanks to Professor Prahlad for helping me sort through all these sources and finding out what I really needed. Oh, it's too far. Across human studies, we've learned that the best use of CRISPR-Cas9 is in hemoglobin metabolism through ex vivo editing. In transfusion-dependent beta thalassemia, the exocell treatments yielded around 91% transfusion independence, and in sickle cell disease, around 97% of patients are free from severe vacuo-occlusive crises for more than 12 months. And leukemias and lymphomas, CRISPR mainly enables better cell therapies rather than directly rewriting malignant genomes. By using precise TRAC locus CAR insertions and by checkpoint knockouts with early multiplex edited T cells, that's how they managed to do that. CRISPR is a potential solution to an age-old problem, but it does have its fallbacks. As of now, the price to use CRISPR-Cas9 to cure disorders such as SCD is around $2.2 million. And keep in mind, there's only around 1 or 2 companies which do have a viable FDA-approved, regulated solution. And discounting the price aspect, the biggest fallback against genetic editing comes with the ethical considerations of modifying the living code of beings, human beings and other animals. So these sort of considerations are what limit the use of CRISPR-Cas9 in the modern day world. And while CRISPR-Cas9 does show promise in monogenic disorders, there is still much work to be done to improve the technology and to make sure that it's accessible for everyone. And for use in polygenic disorders, further development in editing the non-tumorous cells to explore more pathways of treatments for these complex disorders need to be done. CRISPR-Cas9 has the potential to change the world, but it's up to us to make sure that that happens. I want to shout out Professor Prahlad, K. Diaz, and Virgil Torremocha for the help. Thank you to Professor Prahlad to catching me up through speed, even though I was gone for a lot of days, and through my sickness as well, to make sure that I was going strong with my research paper. And thank you to K. Diaz for making sure I knew how to go write a research paper, because I've never written something like this before. And yeah, thank you for your attention. And these are my sources. Yeah. Any questions?
All right. Thank you so much for that presentation. Now, we'll proceed with the comments, suggestions, and of course, questions from our panelists. Let's hear first from Professor Prahlad.
Hey, Shriyans, excellent presentation. Congratulations.
Thank you.
So you mentioned one thing caught my eye there, probably everybody, is the cost of these treatments, right?
Yes.
Ex vivo. So what do you think the potential solution around it? So how do you mitigate it? Did you find
The
anything?
potential solution, right? So
Yeah.
as of right now, I think Exogevi, of Vertex Pharmaceuticals is the company that's developed that treatment, Exogevi, and I think since it's the only product of its kind in the market right now, they have the freedom to go price it as much as they'd like, and they've chosen 2.2 million. And if you also consider the technological aspects of it, it does cost quite an amount to go use CRISPR-Cas9, but I do believe with all the interest and focus surrounding it, and the next couple or so years, we'll be able to see more companies out in the market who produce a similar sort of solution. So, there's going to be more products out there. So the price can decrease quite a bit because there's not going to be a monopoly on it anymore. And if you and with new technologies, the cost of using CRISPR-Cas9 to go treat sickle cell diseases and beta thalassemia might be like reduced a tiny bit.
So one suggestion here is like ex vivo is still going to be like, because it has to be customized, right? That's why it's
Yeah.
very expensive. The people are going towards in vivo gene editing. Okay, so where you can have like off the shelf CRISPR Cas9 tool, the cassette, and you can give it in vivo so that the editing takes place inside the body, so you don't have to customize the treatment. So you may want to look at that a little bit. Okay.
Okay.
Great job.
Thank you.
Congratulations.
Thank you, Professor Prahlad. Professor K.
Hi, Shriyans.
Yeah.
Okay, listening to you earlier, you said about the potential of this study.
Mhm.
How do you think, or do you think this research experience or this research topic has a significant effect on your future academic endeavor or career goal?
Right. So, I've always been interested in sort of like a research-focused medical future, and I do think that CRISPR, when I was back in middle school, that's when it blew up, and everyone was talking about, oh, genetic editing is the future. Genetic editing is how we're gonna, are humans gonna like evolve? And so I think that by studying CRISPR-Cas9 and by studying its effects, I'll be able to become more acquainted with, this new tool, this new this new frenzy that everyone's like been in, and by doing this like early in, I'll be like ahead of everyone else by because I already have some sort of background information in the subject itself. So I do believe that this like base knowledge will help me as I write more papers and I do more work in the future regarding to CRISPR-Cas9.
That's very good then. So good luck with that. Congratulations.
Thank you so much. All right. I believe that Sangeeta will be able to, do a great
Uh,
work. Oh,
I
is it
do have a little
Korean?
something. It's Shrion's. Yeah, I think I've mentioned this like a lot of times. I think somewhere when my mom was signing up, she used her name instead. But
Oh,
yeah.
I see. My bad. Koreans.
Oh, it's all
Okay.
good. Yeah.
Sorry. Okay. Well, of course, Shreyan's congratulations for a job well done. Your attention to detail was remarkable, and it's clear that you really invested time and care in making sure that your work was precise, and it speaks, volumes about your dedication as a researcher. So, congratulations. Let's give Shreyan's again a virtual clap. Congratulations. Job well done. Okay,
Thank
so
you.
you're welcome. We still have two more presenters, and next in line is Ritvik. Ritvik, are you ready?
Yeah, let me just pull it up real quick. Can you see it? Okay.
Yeah.
Wait, can you see the screen? Okay. So
Yes.
this is AI-powered liquid biopsy and how it can be the future of detecting lung cancer early and how it can be used to reduce lung cancer-related deaths by Ritvik, me. I'm a freshman at Acton Boxborough High School. So yeah. So what's the problem with diagnosing lung cancer today? And so basically, it's hard to detect in early stages because it's mainly asymptomatic and it's like it doesn't really show itself until later stages. And because of this, it's honestly like a hassle to like diagnose it and all that. So a lot of people tend to divert from it, especially people who are high risk, like have a history of smoking in their family or contracting lung cancer. And diagnosing, it can also be inaccurate and lead to like false negatives, so people might actually have it and they never find out. And this leads to 75% of diagnosed lung cancer cases are already reached like stage 3 or 4, which at that point, survival rates will drop really like drastically. And when that happens, treatment plans become expensive, time-consuming, and incapable. Basically, they become like less effective, and at that point, it's sort of already too late to change anything, and it becomes really hard to treat. And this actually contributes to why lung cancer causes the most cancer-related deaths in the world. So my solution to this is, oh, sorry. So first, this is my literature review. So I looked at studies that show liquid biopsy and like studies that come like when combined with AI make like really effective ways to diagnose lung cancer. And while I was researching, there were a few gaps, such as like the fact is that liquid biopsy on it by itself, it's already a really new like emerging technique, and that combined with AI, there aren't really many real world cases or studies done on how it can be used. And I use key sources such as clinicaltrials.gov mainly and PubMed. And for my methodology, the study is a comprehensive review, so no like actual studies were done. It's just a review of already existing studies. And I primarily used articles within like the last 10 years or mainly 2019 and 2024 in databases such as PubMed, clinical trials, obviously. And I only found 17 studies, and in those 17, the six most relevant were used. I excluded the other studies because they either didn't like focus on liquid biopsy or they just simply didn't talk about how it can be useful at all. So first, let's go over why and how liquid biopsy works. So what happens first is a liquid sample is taken from a patient. So they'll take any like blood or any bodily fluid for examination. And after that, the actual examination happens where a few tests are run in like labs and they recognize ctDNA or CTCs. So what CTCs and ctDNAs are basically when there's a tumor inside the body, um, like parts of the DNA of the tumor will break off, and that causes ctDNA to form and circulate through, which is why it's called circulating tumor DNA. And then there's also like parts of the tumor itself, so known as CTCs or circulating tumor cells, and those are also circulating through the body, and basically those are like detected, and like you get like amounts or like concentrations of ctDNA or CTCs in liquid biopsy. And finally, the last step of liquid biopsy is the data of like all the, values of ctDNA or CTCs. It's all analyzed and then a result is delivered. So results and discussion. Basically, this is like a table showing normal imaging techniques or like, modern day diagnosis techniques. And then this is how liquid biopsy, which on its own is really new, can work without AI. And this is finally a column showing the advantages of liquid biopsy when combined with AI. So if you look in the normal imaging techniques column, oh yeah, by the way, this table, it shows sort of like how effective these are compared to like benchmarks and diagnosing. So like there's the time to analyze data, like pattern recognition, because patterns can be used to like just be like, oh, this pattern means that there's probably a tumor in the body. And then this is the accuracy of each and every technique. And finally, this is the precision, because when it comes to like getting data from all these techniques, they tend to give like really small and intricate values that can be overlooked. So when normal imaging techniques are used, all of these benchmarks are sort of held back by like, human speed. So especially with liquid biopsy without AI and normal imaging techniques, humans, it could take like a really long time to look at CT scans or look at liquid biopsy data, and that can take a while to happen. And when it comes to pattern recognition, humans are good at pattern recognition, but it's just not as efficient. And sometimes we can recognize false patterns, which could lead to like, false results. And finally, accuracy. We are prone to like errors when you're looking at certain data values. So this can cause more false positives and negatives. And finally, when we're looking at like really intricate CT scans or intricate images, or we're looking at small amounts of data, it can cause us to be like less precise and also cause more errors and accuracies. And most of these are like mainly eliminated when you add in AI with liquid biopsy. And so when you give AI like many sets of data or many like hundreds of sets of data from patients, AI can compute all those within like seconds or minutes. And that's basically because it's a computer program, so it's not held back by human like calculations. Finally, AI is also like really good at pattern recognition, like that's sort of the basis of how it works. So when you give it data from liquid biopsy, it can easily detect patterns and see things that a human would probably miss really easily. And with accuracy, in many cases, AI is like tends to be really accurate, and even when it's not, you can always rerun data sets through it because it can do it within like a really short amount of time. And finally, when you give it like precision, like when you test it for precision with small data values, since it is a machine, usually it will always like compute properly, and it can look at really small um things without being held back. Like if you give it small data sets, it can always like work, like compute it and find patterns within that. So in conclusion, when you take liquid biopsy, which is an already new technique, a really efficient, accurate, and effective, and you combine it with like AI in medical settings, which is powerful, fast, can compute really quickly, and it's also really precise. You get liquid biopsy with AI, which can be a new technique used to quickly and accurately diagnose lung cancer in early stage individuals. So thank you. I'd like to acknowledge Professor Kadias because she helped me with sort of finding my topic and finding what to research about. Professor Julie, sorry, because he helped me sort of write my paper and helped me to like find sources and like the outline. And thank you for Professor Virgil Torremocha for helping us with like presentation and what to do for our presentation and sort of helping us like prepare. And thank you to all the other Gifted Gabber staff that helped me throughout like my journey with making this paper and stuff because I came through like a lot of like issues with scheduling and all that things. So thanks to all those people. And yeah, that's it.
Thank you so much for that presentation, Ritvik. Your confidence and clarity were inspiring, and it's evident that you really understand the heart of your research deeply. So, congratulations, job well done. Now, let's hear the comments and suggestions of our distinguished professors. Let's start first with Professor Prahlad.
Hey, Ritvik, great job. Congratulations.
Thank
So,
you.
yeah, you put very clearly how AI can help. So, but did you find any specific example of a liquid biopsy AI in some cancer that has helped in like better prognosis or better therapy?
So I haven't found like it being really used like in actual practical settings, but I did find one study where they tried using liquid biopsy and AI, and it was a POC study, so it was done like point of care study. So I think that's sort of like the only example I could really give you. But I think if people like more places were to incorporate this technique and like use it with actual patients, then maybe there could be more examples.
Awesome. Yeah. Congratulations. Great job.
Thank you.
Thank you, Professor Prahlad. Professor K.
Hi, Ritvik.
Hello.
I know this one of the most challenging paper that was proposed this time to me was yours, and you're a freshman. What made you decide, by the way, to pursue? I mean, I tried to dissuade you because of the challenge of looking for relevant papers on your study, but you really wanted to do this one. Like, why the interest? Make me understand.
Because I feel like, I mean, me personally, I've always been interested in like how AI can be really helpful. And while everyone is like deciding like, AI for coding and all that stuff, I also have like a really deep interest in medicine, and that combined with like the use of AI, I was thinking like, maybe AI could really be useful when it comes to these sort of things, like replacing human researchers, like not fully replacing, but like being a really efficient alternative because of how it's not held back by like, like how I mentioned like precision errors and all that. So we're like really fully utilizing its power for like helping people. So since this is a topic about that, it really interested me.
Okay, so how was your experience looking for relevant papers? I hope it
I
did
mean,
not.
it was a little hard at first, but I found it like it was really interesting seeing like while I was looking for these papers, I also found many other papers about liquid biopsy. I mean, sorry, like lung cancer as a whole, and it really was like an interesting experience because I've never really done anything like this. So it's like a new experience to me. So that was good.
Okay, I hope you will push forward. I like your dedication. Okay, congratulations on that note alone.
Thank you. Should I answer the question in the chat? Or...
Yeah, thank you. Yeah, you can later answer the questions in the chat box. Okay, you can also type in your answers there. All right.
Okay.
So yeah, thank you again, Ritvik. Let's give Ritvik a virtual clap. And I think Ritvik will be the last presenter for group one. Okay. I'd like to congratulate everyone for a job well done. Thank you for your wonderful presentation. And before we end the presentation for the first batch, I'd like to invite Professor Prahlad and Professor K if they want to share something, maybe a message to our presenters.
Yeah, as I always say, like most of these students, I see all of them actually have almost reached to the point of like publication. So don't stop here. Push it forward. Submit for publication. We just have to go through 1 or 2 rounds of just refining your articles. You're almost there. Let's do it. Okay.
Thank you so much, Dr. Prahlad. And yeah, Professor K.
Yes, I agree. These students have been very dedicated in working on their papers, and they can see how Professor Prahlad and I, thank you, by the way, Professor Prahlad, for being so dedicated and looking into their
Same here.
papers.
Thank you.
It's been an easy task because, like, content wise, I really don't understand. I've always been very open to the students, like, you have to make sure that for your professor, or you get to discuss certain things with your professor. It's like, thank you for addressing the gap. And yes, students, thank you for being so dedicated with your work. Yes, we're almost there. Hopefully you move forward even after this presentation. Congratulations.
Thank you, everyone. Thank you, Professor K. The good thing about Gifted Gabber is that we have our research professor who takes a look at the content, and we also have the creative writer who also, checks the format, the style of the paper. So, we're grateful that you're here at Gifted Gabber and you're really guided by these experts, right? So, of course, let's give our research professor and creative writer a virtual clap. Let's give them a virtual clap. They deserve, the high praises. Thank you so much for your mentorship, and of course to the parents, for your unwavering support. And we'll look forward to seeing the final versions of your research papers, which will mark, of course, the culmination of this rigorous yet rewarding program. So, congratulations to everyone, and we wish you, continued success in your research journey. And before we end, maybe we can have a photo op. I'd like to request everyone to open your camera. Let's make this presentation memorable. Okay. All right. Okay, we're waiting for others. Okay, yeah, that's great. Okay, at the count of 3, show me your best smile. 1, 2, 3. Okay, perfect. Thank you so much, everyone, and we'll see you for the one-on-one session. All right, so that concludes our first batch of presenters today. I'll see you later. Bye.
Thank you.
Thank you.
Thank you.
Okay, so what time are we meeting again? 8 o'clock in 15
Eight-
minutes?
8:30. 8:30.
8:30? Okay.
Yes.
Okay. All right
I'll
then.
Yeah.
have.
Stay safe. Bye.
I'll see you later.
See ya.
Yeah.